So thanks for this great opportunity. Thank you, Sarkis, for the introduction, and thank you, Pramila, for inviting us and for supporting this work. So this is slide 2. Let me go back to slide 1. Oops, it's going the wrong way. So basically, the project that we're funded to do is connecting rare genetic mutations in autism to common biological pathways. And Matt Lally and I will kind of share the responsibility. I'll talk about the past, what we know about rare genetic mutations. And then Matt will talk about his efforts and our efforts to put these rare genetic mutations into common biological pathways. This is supported by the Brain Foundation. We do not yet have data, but Matt and team are moving very quickly, and we're actually on schedule to finish everything as proposed. So just taking kind of a long view of rare genetic mutations in autism, I'll kind of highlight the work of the Autism Sequencing Consortium, which really started with the last stimulus package in 2009, the American Recovery and Reinvestment Act by these two gentlemen here who signed it off. One retired and one came out of retirement. And a small amount of that stimulus money went to the NIH for some crazy ideas. And one of the crazy ideas was whole exome sequencing, which was a technology that, or whole genome sequencing, that was now really becoming available academically within reasonable prices. This was the first unofficial or official meeting of the ASD in 2010 in New York City. We brought a lot of the funders and main labs who were doing sequencing together, and what was going on in 2010, the papers came out in 2012, was whole exome sequencing was supported by the ARA Act, and several labs, 5 labs, decided to work together with ARA funding and sequence, a huge number of samples in those days, 50 trios a laboratory, and put all the data together and see what we saw. At the same time, the Simons Foundation was doing something pretty similar, funding 3 laboratories to work on the Simons SSC samples. And so over the course of 2012, there were these 4 papers had about 1,000 ASD families and focused on de novo or recent mutations in autism. And collectively, if you analyze it jointly, there were 9 genes that were shown to be contributing to autism in a very significant way, by which I mean, if somebody had a mutation in that gene, it was probably contributed to most of their developmental phenotypes. And based on that, something called the hidden species problem, a simpler version is the birthday problem. But basically, you could estimate with 20,000 genes in the genome, how likely is it that you're going to find 9 genes currently hit by mutations out of the greater number of autism genes that should be in the genome. And at that time, in 2012, the data was pretty consistent and hasn't really changed, that about 1,000 genes in the human genome, 1,000 out of 20,000, can be changed in such a way to contribute to autism risk. So in a certain sense, we all have autism variants, and because 5% of our genes are directly involved in some of the processes that underlie the social manifestations of typical development and atypical development. So the argument basically, and just to make it clear, if there's 1,000 genes, right, and we had finished 1,000 cases in 2012, we're far away from discovering all the genes that we can, right? We're at the very early stages. And so the argument of the ASC was, let's put together all of our data prospectively, not publishing small findings at a time, but try and get all the samples run concurrently, which was a bit of a stretch in those days. It was the first example of its kind, really, of large-scale prospective data sharing and sample sharing. The first paper came out in 2014 and now had 22 genes that were autism genes, overlapping with many other developmental disorders. SHANK3 you just heard about. I won't talk much more about that, but that's a good example of a gene where the mutation contributes to the phenotype: autism, intellectual disability, developmental delay, epilepsy, and obviously also GI abnormalities. The most recent paper from the ASD, from the Autism Sequencing Consortium, came out last year, and now it has 102 genes all across the genome, and there's SHANK3 again, that each one, when mutated, in that individual contributes enormously to the risk for autism. And what's gone on to bioRxiv, so this is kind of what we look at. The total sample size is in the tens of thousands of samples and 12,000 cases, individuals with autism. And each one of these funny looking conglomerate of letters is the name of a gene. Of the 20,000 genes of the genome, here are 25 or so that, you know, a fraction of individuals with autism have mutations in that gene. And here's another group of genes where a smaller fraction have mutations in that gene. There's SHANK3, which you just heard about. And so, you know, obviously, if you are working on SHANK3 as an example, you have a biomarker, for lack of a better word, where you can find people that have a mutation in SHANK3 and study them directly. And you can make a mouse model, as Sarkis told you, that has a mutation in SHANK3 and understand what's going on in that mouse, which reflects what's probably going on in patients with that mutation. So you can take basically a rare disorder or orphan disorder approach to these genes and say, I'm not studying autism, or I am studying autism, but I'm studying the autism that's caused by mutations in SHANK3. The most recent analysis by the ASC now doubles the number of cases, 20,000 cases, more from the ASC, but also many more from the Simons Foundation. And we now have 71 genes at a very, very strict threshold. But at a kind of the typical threshold we tend to use, we now have 250 genes that contribute to autism risk. So remember I said there's about 1,000. So, you know, we're honing in on 25%, which is pretty impressive. And this is the genes listed by basically by evidence of their association. There's SHANK3 you just heard about. ADMP is one of the genes. We study SHANK3, ADMP, FOXP1, a couple other ones. So you can select these genes here and approach them, as I said, as a rare disorder. This study really was, I think, another important step in gene discovery in autism. And it's really been led by a conglomerate of individuals who've done an amazing job of bringing the SPARC data and the ASC data together, and also bringing in other kinds of variation, including copy number variation in the analysis. So that should be online. I think it was submitted to bioRxiv on Friday. It should come live in just a couple of days. So, you know, when I started this business of autism discovery, gene discovery, you know, we thought maybe there'll be a couple of genes. And I remember very well when the paper came out and said that there are going to be 4 or 5 genes underlying autism based on how it looks in families. And the idea was that if we could find the gene, the autism gene, or even those 4 or 5, you know, they would highlight a single pathway, right, out of many one, right? There'd be 4 or 5 genes, strong genes. There'd be one pathway, and we'd know where to go for therapeutics. We can talk about this at length, but first of all, we've gone from 4 genes to now 1,000. And every time we look at those genes, there's clearly not one pathway, right? You know, there are neurons, and Sarkis has talked about some of the indirect effects of a neuronal gene on oligodendrocytes. There's lots of ways where you can disrupt the brain to cause behavioral abnormalities of various kinds, including deficits in social behaviors and the presence of repetitive behaviors. So, you know, what I've already kind of intimated is that a lot of groups scientifically are focusing now on a gene by itself. So out of many, E pluribus multis, right? So instead of saying we treat autism as an entity, we could treat autism as a series of independent disorders, right? So your therapeutic focus is on gene-targeted approaches. You think about the treatments that are going to help somebody with a SHANK3 mutation or Phelan-McDermid syndrome. And it may or may not help the next person over. It may, but it also may not. So it's kind of what we do in cancer now, right? And, you know, this is from an epidemiological study in Sweden. if you just take current genetic knowledge, you can actually make a genetic finding in about 28% of individuals with autism. So, there is a path forward for, at least this slice of the pie to say, hey, we can take a rare genetics or orphan disease approach to autism. Of course, there's another larger group that has no genetic diagnosis. So we will make more genetic diagnoses as the number of genes increases. But there's also going to be a significant number that are not going to have a formal mutation that explains much of the phenotype. So this is very kind of where biotech and certainly academia is in autism. They're thinking about a lot of these genes like SHANK3, ADMP, POGZ, SNC2A as rare diseases that can be kind of approached with the orphan drug perspective. And there's a lot of incentives to orphan drug development. It is probably one of the most robust areas of therapeutic growth right now, at least in higher income countries. Some of it has been gene therapy. Some of it's been just remarkable just in the past couple of years. Kids who have certain kinds of developmental abnormalities that will end in their very early death can now get a single injection in the spinal cord and actually so far, certainly be doing very, very well, right? So the idea of gene therapy or gene targeted therapy has really come into its own when people are realizing that it can make profound differences in developmental disorders. Obviously, these ones that we're talking about are more severe than autism, but if the treatment or the intervention is safe, and the individual has, a very difficult trajectory, no language, severe anxiety, severe attentional deficits, low IQ, you begin to think about interventions to kind of to reduce some of those disabilities. Right? So I mentioned that we take this approach of rare variation in autism, and we do a pretty deep dive on 5 genes. SHANK3, ADMP, FOXP1, DDX, which is on the X chromosome, and CHAMP1, which actually comes from another study. We won't go into this, but CHAMP1, ADMP, some of the others actually do form a complex and act together, but others do not, right? And what we try and do with each of those genes is we try and do in vitro work. We try and work on iPSCs or stem cells. We try and look at genetically modified rodents, and we also see patients. So Sarkis was nice enough to talk about some of the genetically modified rodents of SHANK3. We've made a couple, not the one Sarkis talked about, but he mentioned the work of Drew Corelli, which is one of the mice that we've made. But we've also seen a lot of patients with this mutation, and a lot of the phenotype, not only the autism phenotype, but the other behavioral and other phenotypes in Phelan-McDermott syndrome have been carried out by groups like ours that bring in these individuals who really aren't known medically until you bring in a good number of them. And that you also learn something about endpoints for clinical trials. You can develop patient biomarkers, you can look at the mice, and you can do stem cell work. And so this idea that maybe, if you look at the mice, as Sarkis said, you'll learn something about the phenotype and interventions that you can then maybe use for a clinical trial. Another possibility is you use in vitro work like stem cells. You try and find agnostic or gene-targeted approaches. You see if they work in your stem cells. You can make your model. You can try them in your mouse, and you can ultimately do clinical trials there as well. So that's a pretty robust direction the field is going. This is one example of in Phelan-McDermott syndrome, actually, where some deficit we saw in the mice, both in the brain function and in motor abnormalities, suggested a certain approach, and we saw some interesting effects of a specific growth hormone, a growth factor in patients with this disorder, which we've now submitted a follow-on study. However, as good as all that is, we found 250 genes out of 1,000, and many people will not have a mutation. They might have genetic risk, but not a formal mutation. So this approach is only going to get us so far in the autism spectrum. So the other approach would be not out of many, one, or out of many, but e pluribus paucis, which means out of many, few. Can we take 100 genes and come down to just 10 pathways? And instead of thinking about treating each gene mutation independently. We think about treating each of those 10 pathways, depending on the individual, and that individual may not even have a mutation. They might just have a deficit in a pathway that other people with autism have as well, and that would be the point of intervention. So it is this pathway targeted approach. So then the question becomes, how do you get from these rare genetic mutations to actually find common biological pathways? And that is really the subject of the funding. And Matt is going to take it from here. So Matt, are you going to share your own screen, or do you want me to just advance?